Hello, good morning to everybody. I'm Dr. Sivaraja Ganesan from CSA College of Dental Science and Research. Today, I'm going to discuss about the viability of sperms. My aim of the study is to find out the best washing method for those patients having a low count and low motility after semen processing and cryopreservations. So I'm going to use two important stains to assess the viability. Number one will be the Acridine orange and Ethedium Bromide stain, and number two will be the MTT test. I'm using the two tests for testing the viability of sperms. In the current scenario, the routine semen analysis deals with concentration, motility, and morphology, but it provides a little information about the sperm fertility potentials. So, in the assisted reproductive technique, the viability of sperm is an important major concern because after cryopreservation, the many changes will be occur, such as membrane abnormalities, apoptosis, and even though necrosis. So, my current study is designed to evaluate the viability, necrosis, and apoptosis, especially after semen processing and cryopreservation of those patients having a low count and low motility. Now let's see the methodology. First, I'm going to collect the semen sample from those patients having a low count and low motility, and then undergoing the process of semen processing. The semen processing is nothing but we are going to separate the motile sperms from the seminal plasma. So by three important methods. Number one will be the swim-up method. Number two, density graded method. Number three, magnetic activated cell sorting method. So after processing, I'm going to do the cryopreservation for the period of 3 months. After 3 months, I'm going to do the process of thawing. After thawing, I'm going to evaluate the apoptosis, viability, and necrosis. First, we'll see the swim-up method. The main principle is the motile sperms will be swim upwards against the gravity. Now, this is the protocol. First, in the centrifuge tube, I'm going to cut the semen sample and also sperm wash media. And get mixed with, allowed to centrifuge for 2000 RPM for 20 minutes. After centrifugations, we can see the pellet at the bottom of the centrifuge tube. And then I'm going to remove all the supernatant solution to the pellet. I'm going to add the 0.5 ml of sperm wash media and going to keep it in the incubator for 1 hour. After 1 hour, I'm going to aspirate the supernatant solution and store into the cryovial and label it as a swim-up method. And next, we'll see about the density graded method. Here, I'm going to separate the sperms based on the density. So here I'm going to use the two different colloidal silica. Number one will be the 80% of colloidal silica. Number two will be the 40% of colloidal silica. So to the centrifuge tube, I'm going to add both colloidal silica. To the above, I'm going to add the semen sample. And then I'm going to do the centrifuge for 2000 RPM for 20 minutes. After centrifugations, we can see the pellet at the bottom of the centrifuge tube. I'm going to remove all the supernatant solution to the pellet. I'm going to add the 0.5 ml of sperm wash media. I'm going to keep it in the incubator for 1 hour. After 1 hour, I'm going to aspirate the supernatant solution and store it into the cryovial and label it as the density graded method. Next, we'll see the magnetic activated cell sorting method. So here, the final solution, which is performed in the density graded method, transfer into the test tube. That is a 6 ml test tube. To this, add annexin 5 and also binding buffer solution. I'm going to keep in the room temperature for 1 hour. And then I'm going to insert the test tube into the magnet. Here, I'm using the magnet to separate the apoptotic and non-apoptotic spermatozoa. After 1 hour, with the help of 1 ml syringe, I'm going to aspirate the non-apoptotic spermatozoa and transfer into the 6 ml tube and label it as a magnetic activated cell sorting method. After washing, I'm going to do the cryopreservations. For cryopreservations, I'm going to add equal volume of the sperm-freeze media to the cryovial and store into the liquid nitrogen for a period of 3 months. After 3 months, undergoing the process of thawing. After thawing, first I'm going to do the stain, that is Ethedium Bromide and Acridine orange, or you can say the dual staining. So after thawing, to the 96 well plates I'm going to take, I'm going to add the semen sample, that is a washed semen sample, approximately we have the 5 million pounds. And then I'm adding the 10 microliter of Acridine orange and Ethedium Bromide. After that, I'm going to keep in the laminar flow for 30 minutes, maintaining temperature 37 degrees Celsius. After 30 minutes, I'm going to analyze the sample with the help of the fluorescent microscope. So when you see under the fluorescent microscope, we can see the 4 types of sperms, or we can see the 4 types of fluorescence. So we can see first we see the green color, that is a green fluorescence. This indicate that is a viable sperms, because this acridine orange is a vital stain. It will stain both live and dead sperms. So in the live sperms, it will emit the green fluorescence under blue excitations. So whereas the yellow spot, what I'm showing that stay this sperms, or these are the early apoptotic sperms, and this what I'm showing, this is the orange fluorescence, this is the late apoptotic sperms, and the red color what I'm showing, this is the necrosis sperms. So these are the 4 types of sperms will be seen under the fluorescent microscope by using the acridine orange and Ethedium Bromide. So when you see the statistically, I'm going to use the you can see I'm going to use a one-way and a multiple comparison between the groups are done by the quickest test. So you can see the density gradient method shows highest viable when compared to this loop, and also magnetic activated cell sorting method, they are highly statistically significant. Similarly, the necrosis, that is will be seen very low in the density gradient method when compared to this loop and magnetic method, they are highly statistically significant. Next, we'll go for the MTT test. So this MTT test is nothing but it is a dimethyl thiazole and diphenyl tetrazolium bromide test. So this assay is mainly used to determine the cell viability. So this is a calorimetric assay, or you can say it is a very sensitive and it is a quantitative and it's a reliable test. So this test is developed by the Moscow the year 1980. So here what I'm using, those sperm cells having a live cells having a healthy mitochondria. So this healthy mitochondria having an important enzyme that is called as mitochondrial dehydrogenase enzymes. So when we add the MTT test, MTT reagent to the sperm sample, semen sample, they form the formation crystal. So the formation crystal will be seen only in the live sperms. After a formation of the formation crystals, I'm going to add the DMSO, this is a dimethyl sulfoxide, to dissolve the formation crystals. So the amount of formation is produced is directly proportional to the number of viable cells in the semen sample. So this dissolved material is measured spectrophotometrically. So we can see the statistically the data are analyzed by the one-way ANOVA, and multiple comparison between the groups is done by the Tukis test. You can see the density gradient method shows 53% when compared to the Sperm and magnetic activated cell sorting method. So my conclusion is density gradient method was a better washing method for those patients having a low point and low motility based on the cell viability and also apoptosis. So thank you. Thank you very much.